Good afternoon, everyone. Thanks for being here, and thanks to the organizer for inviting me in this interesting meeting. As he already said, my name is Alessia Covre, and I'm a research scientist at the Center for Immuno-Oncology at the University of Siena. And today I will present you how we try to, in our group, we try to improve the clinical outcome of patients, of cancer patients with brain metastases. Well, around 200,000 cancer patients were diagnosed with brain metastases every year in USA. And the overall survival of this patient is very challenging and depends primarily from the histology of the primary tumor. And in this context, more than 70% of melanoma patients develop brain metastases during his life, with the overall survival, median overall survival of around 5 months. Due to the wrong idea that the brain tissue is an immune excluded tissue, and that the drugs cannot cross the blood-brain barrier, the majority of these patients are excluded from clinical trial. The initial change of this scenario came with the advent of immunotherapy, and in particular with the use and then the development of immune checkpoint molecules that target specific. Let me check. Specific molecules on the T cell surface, and with the use of these monoclonal antibodies, we can unleash the brake of immune system, and so improve the immunogenicity of system, so improve the activity of immune system. So with the advent of immune checkpoint inhibitors, as you probably well know, we have dramatically changed the outcome of melanoma patients and cancer patients in general. And we can reach almost the 50% of melanoma patients can have a very long overall survival. In particular, with the use of ECI combination, so the double blockade of two immune checkpoint. So the first acknowledge that the activity of immune checkpoint inhibitors can have, so they can have activity also in brain metastases, comes from two different studies. The first one is one phase 2 study in which melanoma patients with symptomatic or asymptomatic metastases were treated with the immune checkpoint inhibitor ipilimumab. And as you can see here, there is an initial sign of activity of immune checkpoint also in this particular subgroup of patients. And the other study was the NIMIT-M1 study that was conducted in our center by Anna Maria Di Giacomo, that was the principal investigator of this clinical trial, in which melanoma metastatic patients were treated with the combination of ipilimumab and the chemotherapy fotomastin. And among all the patients that were treated in this combination, there were 20 patients with asymptomatic brain metastases. And as you can see here, there was an initial evidence of efficacy, so 50% of patients responded to this immunotherapy. Following this study, there was other two studies, one from the USA, this one, and the other one from the Australian people that investigate the efficacy of the double checkpoint blockades, or nivolumab plus ipilimumab in melanoma metastatic brain metastases. And also this study confirmed the efficacy of immunotherapy, giving almost 50% of response in patients treated with the combination. In addition to this, this was another clinical study that we have conducted in our center. This is the phase 3 NIBIT-2 trial. Again, principal investigator is Anna Maria Di Giacomo, and this is the study with the longest overall survival, the longest follow-up for patients with brain metastases. And again, in this trial, we could confirm that immunotherapy have efficacy also in brain metastases. Oh, let's go better inside of this study. So melanoma, metastatic melanoma patients with asymptomatic brain metastases were enrolled and treated dichotomized to receive or the photomostin, that is a chemotherapy, the standard treatment for brain metastasis, or photomostin, so chemotherapy plus immune checkpoint inhibitors, anti-CTLA-4, ipilimumab, or the combination of two checkpoint inhibitors, ipilimumab plus nivolumab. And as you can see from the results, there is a clear evidence of efficacy when we use the combination of the immune checkpoint inhibitors compared to chemotherapy alone, that is in blue, or chemotherapy combined with the immune checkpoint. So starting from these incredible results, we thought that almost half of patients do not respond to this therapy, so to immunotherapy. So our group are focusing on the identification of biomarkers that could predict the response of melanoma brain metastasis treated with immune checkpoint blockade, utilizing liquid biopsies. That it's not invasive methods that can allow the longitudinal sampling of samples during therapy. And so you can study tumor evolution with these methods. And in particular, we are focusing on genome-wide DNA methylation enrichment, so the methylation landscape of liquid biopsies of these patients, and I will show you in the next few slides why we are looking at this. And also the levels, we are investigating the levels of different immune, pro-inflammatory and anti-inflammatory cytokines. So, as I said before, why epigenetics? Why DNA methylation? So DNA methylation is one of the epigenetic modifications that are known so far in cancer and that is involved in cancer. Epigenetics refer, for those of you that are not in the field, epigenetics refer to heritable changes in gene expression that are not based on the modification of the DNA sequence, and that can explain how two identical twins with the same identical DNA can develop during life a different phenotype. As I said before, there are a lot of different epigenetic modifications that target different enzymes, and in our group, so I can say in the last 30 years, we extensively demonstrated that DNA methylation and drugs that counteract DNA methylation are the best candidate, the most promising agents to be used in combination with the immune checkpoint, because they target different molecules and they upregulate the expression of different and crucial molecules that are involved in the crosstalk between tumor and immune system, so they can increase the immunogenicity of, in particular, cancer cells, but also immune cells. So I don't have time to go in detail with this, but trust me, DNA methylation is very important in cancer development and evolution. So starting from this point and from the evidence that clinical cell-free DNA methylation is used in oncology also for the treatment tumor progression, we investigated the literature and there is, for example, this work from the group of Garcia, in which they analyzed the methylation of plasma, cell-free methylation, in more than 50 patients with different cancer histotype, and they demonstrated that there is a decrease in the methylation. So patients that has a decrease in the methylation in their plasma can respond better to pembrolizumab. So starting from this point, we are going now to monitoring and to study the methylation of our patients enrolled in the NIBITEM2 study. So patients, again, with the asymptomatic brain metastasis treated with the 3 combination, and to identify a set of specific tumor epigenetic signatures that can help us to identify patients that can respond or not to immunotherapy. On the other side, as I said before, we are also looking for several cytokines that are pro- and anti-inflammatory cytokine. For example, in this study from Niccolò Rossi, they have investigated more than, if I remember correctly, more than 90 or almost 100 cytokines in more than 80 melanoma patients treated with immune checkpoint inhibitors. And they found that 2 of these cytokines, this one and the other one, are upregulated in sera from patients, from non-responder patients, as compared to responder 1. So we decided, from all this data, to investigate these 2 cytokines, and adding also other cytokines that are known for their immune stimulatory activity in sera from our patients. And these are the samples that we have collected from the patients. I will show you some preliminary results on the baseline levels of these patients. Of course, all the patients we have all the clinical outcome of these patients, so it's everything is annotated. So when we analyze the soluble levels of these different cytokine, and as you can see here from this graph, I remember you that arm A is patients with brain metastases treated with the chemotherapy, arm B is chemotherapy plus ipilimumab, and arm C is the combination of the 2 immune checkpoint. As you can see from this preliminary data, we can have we can observe the highest levels of this cytokine in patients treated with the immune checkpoint, double combination or immune checkpoint plus chemotherapy. And when we stratified patients from the response, as you can see here from this graph, you can have non-responder and responder patients for each arm. We cannot observe a significant association of this of the levels of this cytokine with the response to therapy. But when we look at in particular at the immunostimulatory cytokine, the TNF-alpha, we can have an increase in the level of this cytokine in responder patients compared to non-responder 1. So these are patients that are treated with the combination of immune checkpoint. And when we associate the levels of the soluble molecules with the overall survival of the patients, as you can see from this graph, you can have a global improved overall survival when patients are treated with the combination of immune checkpoint, as you can see here in red. But looking at again at the immunostimulatory cytokine TNF-alpha, we can have an improve in overall survival. We have an improve in overall survival in patients that are just treated with the combination of immune checkpoint, but has a higher level of TNF-alpha, higher than the median value of TNF-alpha. So these preliminary results started to give us an idea that TNF-alpha could be hypothetically a cytokine that could be relevant for the for its predictive role. Of course, we are now analyzing the level. What I do? What's happened? Okay, we're going to analyze the level of this cytokine also during therapy, so in samples that are that we collected during therapy, and we also we are going to expand the level, the number of chemokine that we are going to analyze. As I said before, the levels of we are going also to analyze the methylome of these patients, and I'm sorry I cannot show you any result on this because the analyses are now ongoing. And so the goal that we would like to reach is to identify biomarkers that could predict the response to therapy, and maybe also identify patients that can be can respond better to this therapy, and maybe, I don't know why, but this is one of our hypothetical goal, to identify that strategies that could be applied to or combined with immunotherapy to improve the efficacy of immunotherapy also in this very subgroup of cancer patients with a dismal prognosis. Okay, it's finished, and I would like to thank, of course, Professoressa Anna Maria Di Giacomo, that is the PI of this project, and all the clinical department and the laboratory department, and of course Professor Michele Maio, that is the director of the Center for Immuno-Oncology where I work. Thank you for your attention. Happy to take any question if you...
Yeah, if there are some questions, we can give you the microphone. Are there some questions? I have a very short curiosity. I'm not, of course, expert in this field. The measurement or the identification of this biomarker, also for early prediction or prevention, are obtained and measured using a long process of for acquiring the biopsy liquid, or they can be faster to be maybe distributed in a population?
Okay, it depends from the technique that you would like to use, because the methylome, it's quite longer as a technique. You have to isolate the DNA, immunoprecipitate that, so it's quite longer. For the analysis of the cytokine, it's very easier. You have to take the, it's an ELISA. It's an ELISA, so in one day you can have the results. So it's easier.
It is something that could be thought to be portable or performed, I don't say at home, but maybe in other public environments, so that this can last a few minutes or 30 minutes or not? Or in the future,
yes.
could be?
In the future, when we have identified one or two cytokines that could be really important for the as biomarker, I think that it could be also portable in very institutes.
Okay, thank you so much.
You're welcome.
Okay.